Happy Wednesday, anatomy students. Today we're going to be talking about the general and the special senses. That corresponds to chapter 18 in your textbook and lab 25 in your lab manual. For the lecture part, I'm just going to be introducing the general and the special senses and going over your senses of touch and taste and smell. And then lab 25 is all about the eye and the ear, so the senses of vision and the senses that are housed in the inner ear. That's equilibrium and hearing. So in this part, I'm just going to be going over the lecture portion. So our bodies are constantly exposed to sensory information, and that sensory information is called stimuli. Anything that's a change in the external world or in the internal world, that's a stimulus, the singular of stimuli. Sensation is the activation of receptors for a particular stimulus. So it's the process of sensing the environment through touch, taste, sound, smell. And this information is sent to our brains in raw form, and that's where perception comes into play. Our conscious awareness of these stimuli is called perception. And perception is the way that we interpret sensations and make sense of everything around us. So the stimuli are detected by sensory receptors throughout the body. These are these receptor cells that can detect a particular stimulus. There are receptors throughout the body for general senses, and these are located all over the body. And these are things like temperature, pain, touch, stretch, and pressure. So general senses are located all over the body. Special senses are those that are localized to specialized organs. So they have special organs for these special senses. And these are things like gustation, which is taste, olfaction, which is smell, vision, equilibrium, and hearing. So here's our basic model again, showing how the sensory system fits in with the nervous system. The stimulus is something that's detected by specialized receptor cells. In this case, it's photoreceptor cells in this specialized organ, the eye. Those specialized receptor cells detect the stimulus, and they stimulate sensory neurons that pass that information along to the central nervous system, where it gets processed. And often this sensory information, as a result of being perceived, results in some sort of response. In this case, the response is movement along a motor pathway to cause you to drink water. So a stimulus causing a change in a receptor, and that's picked up by sensory neurons and passed to the central nervous system. And we'll be focused mostly on these sensory receptors. Some properties of sensory receptors. Receptors exhibit this phenomenon called adaptation. And adaptation means that with prolonged exposure to a constant stimulus, then they tend to decrease the firing of the action potential. They decrease the rate at which they're stimulating sensory neurons. Tonic receptors are those that show really limited adaptation. So if they're receiving the stimulus continuously, processing the stimulus continuously, then they'll continue to send action potentials. So the sensitivity to the stimulus remains constant. Things like balance receptors in the ear will be constantly firing. Receptors for proprioception, that's your sense of body position, as well as pain receptors. Those are all tonic receptors. Phasic receptors are those that quickly detect a new stimulus or a change in a stimulus that's already been applied, and then they rapidly adapt, so they stop firing. So they produce a response, and then with continued exposure, the sensitivity to the stimulus diminishes, and that results in adaptation. So things like deep pressure receptors are phasic receptors. They help you forget that you're sitting on a chair right now. So they stop firing after a while. They're just getting the same information. Adaptation is another way that you would get used to smells. So this is Pike's Place Market in Seattle, where they sell a lot of fish. If you walked into Pike's Place, then your receptors for the smell of fish would be firing away. But after you stood there for a while, you would get used to that smell. That's because your sense of smell has these phasic receptors. So it's rapidly adapting to that constant stimulus, the smell of fish. That doesn't mean that you totally lose your sense of smell, even though you stop perceiving the smell of fish. If somebody walked by and they were doused in Axe body spray, you would smell them right away. So it'd be a change in that stimulus. Receptors can be classified a few different ways. So I told you that general sense receptors are distributed throughout the skin and they're distributed throughout the organs. Those are things like touch, pressure, temperature. And then special sense receptors are those that are housed in complex organs in the head. You can use 3 different criteria to describe receptors, and that's distribution. Where are they located? And that's just general versus special. The stimulus origin, where is the stimulus coming from? And the modality of the stimulus, what type of stimulus it is. So this first one, receptor distribution, it's just, is it a general sense or is it a special sense? So general sense receptors, they're the ones that are found all over the body. The somatic ones are the ones that we're aware of. So they're found in the skin, the mucous membranes lining the body cavities, joints, muscles, tendons, and these are monitoring a variety of stimuli, including texture, pressure, temperature, pain, vibration, stretch. You have individual receptors for each one of those stimuli. The visceral receptors are those that are in the walls of the internal organs and the blood vessels. These are the same type of receptors, it's just where they are. So these are detecting stretch, temperature, pain, still general sense receptors, but they're now on the inside of the body. The special sense receptors are those that are specialized, they're complex, and they have organs that are located in the head. So smell, taste, vision, hearing, and equilibrium. Receptors can also be classified depending on the origin of the stimulus that they're detecting. So exteroreceptors detect stimuli from the external environment. So these would be receptors in the skin, in the special sense organs. Interoreceptors detect stimuli in the internal organs. So these would be things like stretch receptors in the smooth muscle, receptors for pain, pressure, temperature, inside of the viscera, inside of the organs. This last one is sort of in between these two, so it's not exactly the outside world, but not exactly the inside world either. These are proprioceptors, and they detect stimuli that pertains to body position. These are found in muscles and tendons and joints, and this is how you're aware of what position your body is in at any given time. Lastly, receptors can be classified according to the modality of the stimulus that they detect. So the classification by modality, it depends on what's the agent that the stimulus is. The first type is chemoreceptors. Chemoreceptors detect very specific molecules that are dissolved in fluid. So chemoreceptors would be smell and taste. Thermoreceptors detect any changes in temperature. So you have thermoreceptors for hot and for cold, and these are located throughout the skin. Photoreceptors detect light, so they detect changes in the intensity and color and position of light. These are found in the eye. Mechanoreceptors, they detect changes in the shape of a plasma membrane within a particular cell. So mechanoreceptors detect changes in pressure in touch, in vibration, in stretch. These are found in the skin, and these are found in the cochlea, in the inner ear, allowing you to hear. Baroreceptors are a type of mechanoreceptors that detect pressure changes within body structures. So they might detect blood pressure, for instance, and this is by detecting changes in the shape of the plasma membrane of a cell. The last type is the nociceptor. Nociceptors detect painful stimuli. So they detect cell damage. And these are pain receptors that are located throughout the body. It's good to be able to detect pain because it's a way to detect cell damage and to avoid further damage. So, tactile receptors, they're the most numerous type of sensory receptor. They're mechanoreceptors, and they can react to touch, to pressure, to vibration. We already know about some of these from looking at the integumentary system. You know that they can be located in the dermis and in the subcutaneous layer, or the hypodermis. There are two types, some that are unencapsulated. These are just simple dendritic endings. Remember that dendrites are the receptive part of a neuron. And they can be encapsulated, and these are more complex dendritic endings that are wrapped in connective tissue or wrapped in glial cells. These tactile receptors are considered general sense receptors because they're located all over the body. They could be exteroreceptors if they're on the surface, or they could be interoreceptors if they're within internal organs. So some that we already know about include some unencapsulated tactile receptors, so tactile discs, also known as Merkel's discs, free nerve endings. These are just dendrites that are going throughout the dermis and the epidermis here, and nerves that are wrapping around the base of a hair. That's a root hair plexus, and that allows you to detect when that hair is bent. Encapsulated tactile receptors include a tactile corpuscle, and this should look familiar up here in the dermal papillae. So we called it a Meissner's corpuscle or a tactile corpuscle. Another one that we have seen before is this lamellated corpuscle or Pacinian corpuscle down here right above the subcutaneous layer. So the benefit of these encapsulated tactile receptors is that it takes more pressure, a stronger stimulus, in order to stimulate them as compared to the unencapsulated tactile receptors. So you can detect a very light touch with these free nerve endings, a slightly stronger touch with this tactile corpuscle or Meissner's corpuscle, and a really strong touch if these lamellated corpuscles are firing. Proprioception, it's that sense of movement and of body position. So in order for the nervous system to be able to effectively control skeletal movements, it has to receive continuous sensory feedback from the muscles. And this feedback is proprioception. And it consists of the sensory detection of muscle tension via the Golgi tendon organ and of the changes in muscle length via the muscle spindles. So these are all mechanoreceptors. They're stimulated by mechanical forces that change the shape of the cell membrane. So the muscle spindle helps to determine the length of a muscle, or in other words, how contracted it is. So the muscle spindle is a proprioceptor. It's a sense organ that's receiving information from the muscle, and it senses stretch and speed of stretch in the muscle. When you stretch and you feel that message that you're at the end point of your stretch, then the spindle is sending a signal telling you not to stretch any farther. So this sense organ not only lets your nervous system know how far a muscle is stretched, but it protects you from overstretching. The Golgi tendon organ has these same dendritic endings, but now they're inside of a tendon. Remember that tendons attach muscle to bone. And the Golgi tendon organ determines how much load or how much weight is being exerted at a particular point. So it's a proprioceptor. It's a sense organ that's receiving information from the tendon and sensing tension. So when you're lifting weights or just moving around, the Golgi tendon organ is that sense organ that tells you how much tension the muscle is exerting. If there's too much tension, then the Golgi tendon organ will inhibit the muscle from creating any force and protecting you from injuring yourself. So I said that you can have interoreceptors, these visceral senses, and in this case, it's the same type of receptors, but now they're inside of the body. So you can have free nerve endings inside of the body. You can have Meissner's corpuscles inside of the body, or Pacinian corpuscles on the inside of the body. And this is one way, for instance, that you can tell when your stomach is full. It's because you have these mechanoreceptors in the linings of the internal organs. So these would be mechanoreceptors, and they'd be interoreceptors. And this would be considered a general sense. Said that the perception can be referred to the surface. What that means is that the perception that you're actually feeling on the inside of the body is perceived at a location other than where it's actually being stimulated. So an example of this is, sometimes people say that when you have a heart attack, then you feel pain down the left arm. So that pain that's actually being felt at the heart, then that is being referred to the surface, and it's felt in the left arm instead of in the heart. Another less serious example is the example of an ice cream headache or of a brain freeze. This is another example of referred pain. And this is where you're actually getting pain because of the rapid cooling and rewarming of the capillaries in the sinuses, but instead you feel pain in your throat and in your palate. So that's pain that's being referred to another location. It's referred to the surface. And this is because we have sensory neurons, this afferent neuron line that's coming from the viscera, things like the heart. So we have a sensory neuron, look, that pseudo-unipolar neuron coming in the dorsal side of the spinal cord. So it's an afferent line. And along that same spinal nerve, you have pseudo-unipolar sensory neurons coming from the skin, and they're converging at that same spot on the spinal cord, and they're taking the same path to the brain, so the wires get crossed. When you're processing that information, it's much more common to get a stimulus from the skin than from the heart. So this common line is interpreted as coming from the skin, even though the initial stimulus is really from the heart. So now we're getting into some of the special senses, those that have a special organ located in the head. Gustation is your sense of taste. And for these, you require gustatory cells within taste buds. So gustation takes place on the dorsal surface of the tongue. So here's the dorsal surface of the tongue right here, sticking out. And if you look at the surface of the tongue, you see all those little bumps. If you stick out your tongue in the mirror, you see all those little bumps on top of it. Those are papillae. The papillae, those nipple-like projections, those are not the taste buds. The taste buds are inside of those papillae. So each taste bud is associated with these nipple-like elevations that are called papillae, but the structures that you see are the papillae, not the taste buds. So if you were to look inside of the taste bud, what you see are these taste cells that are illustrated in yellow here, and the supporting cells that are supporting the taste cells. So there's a taste bud, it's this whole structure right here. And the taste buds have a tiny little taste hair, a projection out from the outer end of the taste cells. And those are the sensory parts of the receptor cells. So these taste cells, or gustatory cells, they detect tastins, which are molecules and ions within food. So they have one single taste hair that detects molecules that are dissolved in saliva. And each taste cell is particularly susceptible to one particular tastin. And that's why I say that there are 5 different flavors. You can detect sweet, sour, salty, bitter, and umami. So each one of these taste cells is going to be responsible for tasting just one of those flavors. And then when this taste hair detects that particular tastin, then it passes this information along sensory nerve fibers, and it gets transmitted to the central nervous system, either through cranial nerve 7, the facial nerve, or through cranial nerve 9, the glossopharyngeal cranial nerve. So the taste cells with taste hairs as part of a taste bud in a papilla on the tongue detect molecules that are dissolved in saliva and send messages to the central nervous system through cranial nerve 7 and cranial nerve 9. And this is a chemoreceptor because it's detecting a molecule dissolved in fluid. It's an exteroreceptor because it's a stimulus coming from outside of the body. And it would be considered a special sense because it requires this special organ. Another chemoreceptor is your sense of olfaction or your sense of smell. So chemoreceptors require that something be dissolved in solution. In this case, it's odorants that are chemicals that are dissolved in the mucus of the nasal cavity, and then they're detected. So when you smell something, what's actually happening? There are volatile molecules from that thing that are making physical contact with your body. They're getting dissolved in solution. They're not necessarily solids. They could be volatile gases. So let's look up here at the top first. Here's somebody, they're stuck at home and they're about to enjoy this steaming mug of hot Dr. Pepper. And there are molecules of that hot Dr. Pepper that are traveling through the air and into the nasal conchae. And they're traveling up through the nasal cavity. And they're making physical contact with the olfactory epithelium up here, binding to receptor cells in the olfactory epithelium. Remember, this is right below that cribriform plate of the ethmoid bone. And then that information gets passed along the olfactory tract to the brain for processing. So if we look a little bit closer over here, again, down at the bottom, those are those odor molecules, those volatile molecules of the hot Dr. Pepper. And they're being dissolved in this mucus layer. Extending into the mucus layer are the olfactory receptor cells. The dendrites of the olfactory receptor cells have olfactory hairs, similar to the taste hairs, that stick out into the mucus layer and bind to just one molecule. They bind to a particular molecule and send that information along the olfactory receptor cell, which travels up through the cribriform plate. Remember, this is the olfactory foramina within the cribriform plate. And overlying that is the olfactory bulb, the start of cranial nerve number 1, which passes that information back along to the central nervous system for processing. So when those olfactory receptor cells within the olfactory epithelium get stimulated, they send a nerve impulse in their axons that pass through the cribriform plate of the ethmoid bone and synapse on neurons within the olfactory bulb. And down here is an olfactory epithelium photomicrograph showing the same thing. Here's the mucus layer down here with olfactory hairs. that are specialized to detect just one odorant molecule. And then if they're stimulated, that nerve impulse travels up through the olfactory receptor cells and gets passed to the neurons within the olfactory bulb. And you all know about the olfactory bulb because you've looked at the inferior view of the brain. And if you do look at the inferior view of the brain and you look near the longitudinal fissure or the interhemispheric fissure, and on the inferior surface of the telencephalon, then you'll see these olfactory bulbs that make up cranial nerve number 1. The sense of olfaction is not as sensitive in humans compared to many other animals. So a lot of other animals devote a lot more brain power to interpreting smells. Dogs, for instance, probably have about 40 times larger, relatively speaking, area of the brain devoted to analyzing odors. So if we were doing a sheep dissection in lab, you'd see that the sheep brain is similar to what dogs might have. This whole structure up here is the olfactory bulb. So sheep have a lot of a stronger sense of smell than people do. A lot of animals devote a lot more brain power to interpreting smells than humans do. So our olfactory bulb, relatively small. What that allows us to do is have a lot more of this frontal lobe, a lot more conscious decision making, a lot more analysis, a lot more critical thinking, and that means that the olfactory bulb has shrunk comparatively. So here's the olfactory pathway. Here's somebody, they're smelling not hot Dr. Pepper, but they're smelling sweet rose, and there are actual molecules of odor leaving that rose, traveling through the nasal cavity and traveling up to the olfactory epithelium. There are odorant molecules that are binding to receptors, binding to those olfactory hairs that contain the receptors for just one odorant molecule. And depending on which receptor cells are stimulated, different smells will be perceived. And then those olfactory receptor cells get activated by the binding of that odorant, send electrical signals through the cribriform plate of the ethmoid bone. Those signals get relayed via converged axons. And then they are transmitted to the higher regions of the brain through these axon bundles called the olfactory tracts to that olfactory cortex of the temporal lobe. So I hope that you'll take a minute to stop and smell the roses before you jump into the senses part 2.